think we should start. So, hello everyone, and welcome to the 5th seminar of the Deep Sea Biology seminar series. Today we're gonna have two talks on hydrothermal vent research. A first talk from Takuya Yahagi from the Atmosphere and Research, Ocean Research Institute of the University of Tokyo, that will talk to us about larvae and their ontogenic migration between the vent and the surface water. And then we're gonna have a second talk on hydrothermal vents about the microbial iron mats this time that were found in a new discovered Arctic vent field. And this will be presented by Petra Hribovsek. I'm sorry if I misspelled. And this is she is from the Center for Deep Sea Research in the University of Bergen in Norway. So Takuya, if you want, you can start sharing the screen.
Yes, I will share my presentation slides and. Okay, so can you see this slide? Okay. Okay. Okay, so today, I would like to thank organizing members of the SBS seminars, and so thank you for taking your time to listen to my presentation. And today, I will talk about my current work, about ontogenetic migration of gastropod larvae between deep sea hydrothermal vents, via surface waters, revealed by culture experiments, population genetics, and geochemical analysis of shells. And so before starting my presentation, I will introduce my institute. So my institute, called AORI, consists of 300 people, including faculty staff members and graduate students. My institute takes care of two operations of two research vessels, namely Hakomar and Shinsemar. I often join research cruises using these two research vessels. And so also my institute has a sushi restaurant near the entrance. So if you have a chance to visit AORI, you can enjoy many kinds of sushi bowls every day and so on. So we are very welcome to you. And in my institute, I belong to Marine Bentology Group and of Division of Marine Life Science. And now our ongoing research themes as follows. And especially, I focus on the evolution and the ecology of deep sea benthos, including chemosynthetic-based ecosystems. And so we use ROV and beam trawls to collect these basic animals during such research cruises. And so among deep sea, hydrothermal vents communities are supported by chemosynthetic bacteria and often dominated by molluscs, crustaceans, and annelids. And they show high biomass and endemisms. And so these hydrothermal fields are located on mid-ocean ridge and Backrack Basin, and also underwater volcanoes. So dispersal is important for maintaining meta population among such unstable and isolated fields, vent fields. And so larval dispersal, especially contributes to geographic distribution, population dynamics and evolutionary processes of vent endemic animals. So however, little is known as to the extent that vent larvae migrate vertical to shallow waters and experience strong currents and richer food supplies. So the present study focused on hydrothermal vent limpet Shinkai lepers. The species of the genus has a wide geographic distribution range in lower latitude areas in world ocean. And the genus is an excellent taxon to study larval ecology and dispersal. So because the first, so embryos can be observed in transport and egg capsules. And second, hatched planktrophic larvae can be cultured under atmospheric pressure. And third, settlement size can be inferred from post-conch morphology. So especially, so I have evaluated larval dispersal of Syncarepas myogenensis in the Northwestern Pacific. So the species has a wide geographic distribution, so between Okinawa Trough and Izogasawara Arc. The black boxes indicate bent sites with Syncarepas myogenensis, while white circles indicated bent sites without Syncarepas myogenensis. And so our genetic analysis using mitochondrial CO1 gene for 77 individuals showed high connectivity among all populations. So even they about more than 1,000 kilometers. And also the deposit range is about from 400 meter to 1,000 meter. So I cultured larvae under the atmospheric pressure, and I could observe their vertical swimming behavior of hatched larvae. And so this video also shows their vertical swimming behavior. And so the hatched larvae constantly swim upward. And also the vertical migration from hydrothermal vents to surface was calculated to take 4 to 43 days. And they're very tiny larvae, so swim upward. So I also cultured larvae for 9 conditions and for 180 days. And so our culture experiments showed the best survival and growth of the larvae was shown at sea surface temperature and when fed with phytoplankton diatom. So at the 25 degrees, the larvae most survived and best growth. And on the other hand, so adult limpets of the species inhabit around 10 degrees. No growth or few growth was seen for the larvae at this temperature. And so in terms of settlement size of the species, so based on the protoconch morphology, so the settlement size is about 700 micrometer. So but our culture experiment, so the larvae reached about 300 micrometer for 6 months. So that's why we can estimate their planktonic duration for more than 1 year. So they have very long planktonic duration. And so based on these evidence from culture experiments and population genetics, so the larvae most likely to migrate to surface waters, so where high phytoplankton biomass and strong currents enable their growth and long distance dispersal for over many months. So first time there's an ontogenetic vertical migration of hydrothermal bent animal was demonstrated. And this new finding has also implications for biogeography of bent faunas in high latitude areas. So interestingly, no record has been found for bent animals with planktrophic development mode in Arctic and Southern oceans. So that's why cold surface waters may prevent the larval dispersal of planktrophic larvae. And this indicates sea surface temperature represents an important factor to determine their geographic distribution of deep sea hydrothermal bent animals. And so our following studies have also showed the long distance dispersal for two confamiliar species with a planktrophic development. And so our genetic studies using mitochondrial C1 gene showed the population connectivity along northern mid-Atlantic ridge. And so as a species, so in the southwestern Pacific, and so both more than 3,000 kilometer distance. And so we also have studied, so our study shows permictic population of planktrophic species of bent shrimps in northwestern Pacific, so as well as the gastropod species. So, but to make sure so their ontogenetic vertical migration, so we need to look at evidence from those that actually settle to hydrothermal vents. And so stable isotope and trace element measurements of biogenic carbonates are invariable for reconstruction of autoecology of individuals. So here we first describe the oxygen isotope and the trace element compositions of molluscan shells before and after metamorphosis for the inference of larval migration from deep sea hydrothermal vents. So stable oxygen isotopic composition of shells provide an effective tool for understanding the surrounding temperature throughout the ontogeny of molluscs. So we analyze the stable oxygen isotopes of 33 newly settled juveniles of Shinkai myogenesis and Shinkai kaikatensis from Kaikata Seamount is Ogasawara arc. So where adult habitats, the temperature of adult habitat is 11 degrees and surface water is 19 to 28. And we also analyze the shallow water relatives as a control. And also we analyze using isotope ratio mass spectrometry. And so these pictures show settlement juveniles or Shinkarepas kaikatensis. And so in our analysis, so we use just settlement individuals with only larval shells. So the reason why we use newly settled juveniles is because these individuals, so such individuals, settlement individuals has already grown some adult shells. So such adult shells, so post-metamorphic shell layer, so prevents accurate measurements. So that's why we use only newly settled juveniles without depression inside the protoconch. And also we can take accurate measurement. And so this figure shows the results of stable oxygen isotopes for Shinkai limpets for inferring vertical migration. And so based on this analysis, oxygen isotopes analysis shows that protoconchs of Shinkai lepers have lower oxygen isotope values than the pteroconchs, the lower values. And so while there is similar temperature between protoconch and pteroconchs for control species. And so based on these oxygen isotope values, we first reconstructed water temperature for the larval period of bent animals. And so the temperature during larval periods is constant, So the temperature during larval periods is constant with sea surface temperatures. So that's why we can conclude, all larvae migrate to the surface water as an obligatory part of the species life cycle to feed on phytoplankton. So on the other hand, how about when adult lives in similar temperature as surface waters? So we also analyzed the Shinkai tolmani, from Lao Basin in southwestern Pacific. So in this species, the temperature is similar between adult bent habitat and sea surface waters. So when we measured their oxygen isotope compositions, so they showed similar temperature signals between protoconchs and pteroconchs. So that's why it is so distinguished. So it is difficult to evaluate their ontogenetic particle migration. So based on only the stable oxygen analysis. So therefore, we measured the trace element composition of larval shells and adult shells to evaluate their ontogenetic particle migration. So the trace elements such as manganese and barium are present at very different concentrations in surface waters and hydrothermal fluids. So we analyzed the 3 species of Shinkai-Lepas limpets and also the confamiliar non-bento species as a control. And so samples were embroidered in resin and polished, and we observed under the same. And so this figure shows the samples after trace element analysis using nanosims. So we measured 4 trace elements, so such as magnesium, calcium, manganese, and barium by 5 to 6 micrometer beam spots. And so we found the shell thickness is 8 to 15 micrometer. And so this specimens, so the blue arrows indicate the boundary between pteroconch adult shells and protoconch for adult shells. Yes, this is so boundary. And we compare relative concentrations, so between protoconchs and pteroconchs. And so this figure shows the results of trace element analysis of shells for 3 species of Shinkai-Lepas and a control. And so the value is for pteroconchs and protoconchs for bariums and manganese. And so this, the first elemental analysis for the library migration of bent animals showed pteroconchs grown in bent environments contain much more barium and manganese than protoconchs. So this trace element analysis concordantly supports larval migrations to surface waters, so inferred from larval culture experiments and population genetics, and also stable oxygen isotopics. So we believe that this method is applicable to all bent mollusks, so such as Shinkai-Lepas, Bachmoderas, and also Albiniconcha snails. So in summary, we can conclude all larvae of studied species of Shinkai-Lepas migrate to the surface water as an obligatory part of the species life cycle. And also, our temperature reconstruction methods, and in combination with trace element analysis, enables us to trace the entire life cycles of bent mollusks. And so these studies can contribute to development of ocean circulation models for larval dispersal of bent animals. So for examples, these figures show the larval dispersal models based on dispersal depths at 1,000 meters and planktonic duration for about 80 days. So the evaluation of larval behavior would help develop biophysical models and establish the marine protected areas. So finally, I would like to thank these my collaborative people and funding, and also thank you for kind your attention. Okay, so thank you very much. So please feel free to ask me. So if you have a question.
Thanks a lot, Takuya. So yeah, if anyone have any question for Takuya, you can either use the chat box or just raise your hand and speak. Does anyone have a question?
I have a question.
Okay, yeah.
Leon Hoffman here from Wilhelmshaven in Germany. I saw your talk in Munich, I believe. Yeah, but not with the last end of it, with all the chemical analysis that was. First of all, let me congratulate you. This is tremendous research. Very good. Well done.
Thank you.
I went back to one of the papers of Bergen and Boucher, where he described Divia briandi, and you were involved in that paper as well, if you remember.
Yeah.
He writes there that the animal puts pockets of larvae of about 10 only on the seafloor before they start developing into Veliger larvae. Yeah.
Yes.
You describe a very elaborate journey of these Veliger larvae. Essentially, they go to the surface and they live there for 6 months, and then they find back their way again to a hydrothermal vent. And still, this process is very effective because this animal is very common on the mid-Atlantic ridge. How do you see that if an animal produces so little larvae, essentially per cycle, can be so effective in such an environment, finding back its habitat again after 6 months?
Yes. And so yes, so they have so high fecundity, so for all Syncarepas limpets and also Divia. And so they showed no seasonality for reproductive their reproduction. And so they often so laid many egg capsules, so more than million embryos for adult individuals. And so yeah, so in the case of Divia briandi, so this species also showed population connectivity, so along the wide geographic distribution. And so I have not yet analyzed the larval shells of this species. So yeah, we are very interested to analyze their larval shells to evaluate ontogenetic migration for this species.
Yeah, I guess that they live the same way as the one that you evaluated from Japan. I was, as you said, probably they lay many of these pockets of eggs, so that they continuously lay these eggs, and therefore somehow they keep an effective reproduction going. It's still the question how they find back their habitat, I suppose, but there are many questions that we can't yet answer.
Yeah. So very interesting points. And so fauna, chemical composition and so high temperature and also sounds from hydrothermal vents important factor to determine settlement place for hydrothermal vent animals. I also, in my best knowledge, so there is no such culture experiments to detect their settlement mechanisms for hydrothermal vent species. That's very interesting, I think. So too.
Thanks a lot. Thanks a lot.
Yes.
Super.
Thank you very much.
I think we have another question from Jenny Nehaus.
Yes, hello, Jenny here from Hamburg. Thank you very much for this very interesting presentation. My question was kind of answered already. I just wanted to say that this is very interesting, and I myself work on barnacles, like deep sea barnacles, that have these naupliar larvae. And do you think that they are planktotrophic as well? Do you think that they could exhibit a similar behavior? Just do you think beligers have a higher swimming capacity compared to naupliar, or?
Yeah. So the barnacle belongs to genus Neobelluca, no?
No, it's balanomorphs I'm working on. So it's not the stalked, but the sessile ones.
Yeah. So in my understanding, so so many barnacles, bent barnacles showed less trophic development mode. So there is no planktotrophic, so larval phase. So that's why, so yeah, many barnacle bent species so disperse in deep sea waters. So maybe so so they have so yolk, and so they grow and they can reach their settlement size for using their yolk. So that's why so they may not so vertically behavior. I think.
Yeah, it's correct. It's just in my species, it's actually planktotrophic behavior, so they don't have lasistotrophic larvae. So I don't know. Could you? Yeah, maybe. Yeah, I'm just speculating here, but it's such a cool finding. So I was just thinking that if veligers manage to migrate up in the water column, maybe planktotrophic Nalpli can as well. It's yeah.
So I'm also very interested in universal of vertical migration behavior of bent animals, all bent animals. That's why very interesting point for barnacles.
Yeah, it's really, really cool. Thank you so much.
Yeah, thank you very much.
We still have time for one last question. So there is one question from Jinsen in the chat. Do you want me to read it, or you can just
and yeah, also Pierre, so in chat.
Yeah, so he's saying that so Jinsen from the Ocean University of China is asking, have you ever tried to feed the larva with phytoplankton?
Yes, so actually, so in my experiments, so the larvae so fed phytoplankton diatom, so and growth are the best at sea surface temperature. Yeah, so that's why so we confirmed the so feeding so phytoplankton of bent species, bent larvae.
Okay, thank you. I had myself one question on your experiment of culturing the larva. I think the last curve at 30 degrees is showing something like the best growth is at 20 and 25, and at 30 you observe a depth of the larvae. And since you also showed with your isotope analysis that this vertical migration on the surface is like obligatory for the life cycle, do you think like increase of the temperature of the ocean in the coming years will affect also hydrothermal vents, and that they are not so isolated from climate change?
So it's very interesting point. And so in this in the case of this species, so they can't so survive at so 30 degrees. And so interesting, so their geographic distribution so is consistent with sea surface temperature. Southern so region, so the temperature of the southern region so is reach 30 degrees, but there is no records of Syncarpus myogenesis this species. So that's why so the sea surface temperature so may represent a determining geographic distribution of the species, and so that's why so future climate change so also may affect on their geographic distribution. So even so deep sea hydrothermal vent species so with planktotrophic development mode.
We have one last question. We're going to take one minute extra of the schedule. Leon Hoffman again is asking why all your uploaded network are so wide with so many small mutations, and does it say something about their genetic stability, do you think?
Yes, so my uptime network.
Yeah, I think most
one.
Yeah, this one for example.
Yeah. So I think so generally so they show the high haplotype diversity so compared with other hydrothermal vent species. And
Yeah.
so, so this means, so this indicates, so there are genetic stability, among populations, I think so.
Yeah, it could be something common to many vent species, you think?
Yes,
Okay.
so I think, many planktrophic species shows high habitat diversity. So, based on their genetic stability.
Okay, we're going to move to the next talk about microbial iron mat from a new discovered Arctic vent field, in the Arctic region. Takuya Yahagi mentioned several times some interesting patterns about larvae on Northern and Southern Ocean, Arctic and Southern Ocean. So I think there is maybe some interesting connection to make. So Petra Hribovsek, you can start your presentation.
Okay, great. Let me just share the screen.
We can see your screen.
Yeah, you can see the right one. Okay.
Yeah, working well.
Okay, yeah. Hi, everyone, and greetings from Norway. And thanks so much for having me here. So, and thanks, Pierre and DBS for hosting this seminar. I think it's a really nice meeting place. So I'm a PhD candidate in microbial ecology at the University of Bergen and also at the Center for Deep Sea Research. And one of the things I study are uncultivated iron oxidizing bacteria of hydrothermal vent fields. And today I want to share with you what we have learned at this relatively new vent field in the Arctic and the microbial iron mats there. So I want to take you to our study site. So we know of a couple of hydrothermal vent fields along this Arctic mid-ocean ridge. So this is between, like north of Norway, between this small island of Jan Mayen and like the Svalbard archipelago. And this is where my group does research. And I want to introduce you to fauna vent fields, which is this small red dot. This is a vent field that was discovered in 2018 with the first time our research cruise was going there in 2019. And it was completely different from other vent fields that we've seen before on this ridge. So what we have seen is this tall black chimneys, black smokers that are venting at approximately 250 degrees. And the fluid here is very rich in iron and hydrogen. And you have this microbial mats, this thick orange fluff that is covering the chimneys that you can see. So this unique geochemistry with iron and hydrogen makes this a unique study site for iron oxidizing microbes. So if we want to take a sample of these microbial mats, we use so-called suction sampler. Here you can see one at work, and you can see really like the texture of this mats that get sucked in. And for everything to be possible, we use this kind of ROV or EGE 6000 that we have, and we put it down at approximately 3000 meters. Yeah. But why at all study iron oxidizing bacteria? So there is this thing that iron is everywhere, and we can have iron in soluble form. So we have Fe2, and this one can be both microbially and chemically oxidized into iron 3. This can be very often in form of iron oxides, so like non-soluble, as we can sometimes see in rust. So it would have this rusty color, and you can sometimes see that, for example, on ships. So this process kind of affects us, so like corrosion, for example. And it can also affect other processes and things that we care about, such as soil quality, water quality. And much of this is just because of this redox activity of iron. And iron 3 can also get reduced microbially. This, for example, happens in deep sea sediments in certain layers. Yeah, but then we have these iron oxidizing microbes that oxidize iron 2 to iron 3, and they couple this with breathing oxygen. And there are some certain challenges that come with being this kind of microbe. So the Fe2 to Fe3 redox potential is quite high. This means that there is very low energy that you can get from this process, meaning that these microbes need to really oxidize a lot of iron to get enough energy. One of the things is also that you're constantly competing with abiotic oxidation, which can happen very quickly. So these microbes have kind of adapted to this in a way that they live at very low oxygen concentrations, where they can still compete with the abiotic oxidation. And at the same time, they also fix CO2 into organic carbon. Then we have Fe2 that is non-soluble. It very quickly oxidizes, so it's mostly in form of iron oxides. And in certain bacteria that oxidize iron, they also precipitate these iron oxides in specific structures, like in form of stalks, bifurcating structures, or tubes, like you see on these images. With that, they also kind of surpass one of the other challenges that these microbes have, which is to avoid the incrustation within the iron oxides. So they put it kind of outside of the cell, outside of the way. And so it's in a way an adaptation, but also has been hypothesized that this is, with these structures, a way to like attach themselves and also maybe position themselves in the gradient of oxygen and iron. What we see on these non-soluble iron oxides is also adsorption. That would be adsorption of other metals, such as heavy metals, for example, and phosphate and organic carbon. A lot of these microbes are also involved in nitrogen cycling. So we have these iron oxidizing microbes that gain energy by oxidizing iron, by fixing carbon, cycling nitrogen, and also forming these iron oxides that very strongly bind metals, phosphate, and organic carbon. So they kind of connect by geochemical cycles, and this makes it crucial to understand iron cycling a bit better. But also, so they kind of expand our knowledge in how diverse life could be, but they also maybe could help us with bioremediation, so remediation of metal pollution and recovery of resources. And as mentioned before, some microbes that oxidize iron make these kind of oxide structures, for example, in forms of stalks. And the morphology of these structures is kind of dependent on environmental factors, such as availability of oxygen. So in a way, they can fossilize information on environmental conditions. And these structures can be preserved in ancient iron oxide deposits, like the ones from Precambrian, and that can help us understand the conditions and life of early Earth. These structures can potentially also be used as biosignatures. This would mean that they could be used as a proxy that life has existed on other planets, like, for example, on Mars, that is very iron-rich planet. But coming back to our study, so to study the iron mats at Bergen, we have used genome resolved metagenomics. And we have used both the short and long read sequencing. So we have done the nanopore MinION sequencing on board of a research vessel when we're there. And when we came home, we did the high-seq Illumina sequencing. We have then done a long read assembly that was polished with Illumina short reads. And finally, made the metagenome assembled genomes that were also manually refined. And then we can use those genomes for all kinds of analysis. So we looked at iron oxidizers that were there, compared them between each other with comparative genomics, used phylogenomics to determine the closest evolutionary relationships, looked at the composition of the iron mats, and of course predicted the metabolism based on key enzymes and annotation. And all of this was then also combined with different types of microscopy, such as fluorescence microscopy and scanning electron microscopy. So this is a first look into this iron mats at this study site. And we could see that these iron mats were really dominated by tubular structures of iron oxide, also called sheets. And these are these tubes that you can see on the image that are approximately 2 micrometers wide, just basically as wide to fit a cell inside. And these are produced by a group of iron oxidizers that are called zeta protobacteria that oxidize iron. And indeed, we could also see cells of zeta protobacteria inside of these structures that we have detected using fluorescence in situ hybridization. So until now, we know which zeta protobacteria produce stalks because we have isolates of them. So this is of a very well-known genus Mari profundus, but we do not know who produces sheets, so these tubular structures. This is mostly because cultivation of this group has been quite challenging. So it really requires like good techniques and a lot of time on the hands and cultivation. But from the community analysis, we could see that zeta protobacteria genomes of exclusively genus Cursea were present in the iron mats, also based on the 16S RNA genes and amplicons. So you can see in this phylogenomic tree, you can see the genomes that are marked in orange. Those are genomes of zeta protobacteria that were present in the mats, and all of them are quite closely related and belong to the genus of Cursea. So Cursea might be the one that is producing these iron oxide sheets and forming these iron mats. With that, genus Cursea kind of now would have a larger morphology of iron oxides than previously. But of course, as with all metagenomic studies, this would need to be confirmed with cultivation. So with these structures that kind of make up the bulk of the mat, is really dominating the whole mat, Cursea provides also an environment for other microorganisms. So we can see that zeta protobacteria makes only a part of the mat. The most abundant other members are gamma protobacteria and Campylobacter of the genus Sulforovum. Sulforovum is quite common in iron mat, not iron mats, but in other microbial mats covering black smokers, for example. And what we could see, also we could identify some high quality viral genomes. And those genomes were predicted to have Sulforamonas and gamma protobacteria as a host, both of which are quite abundant in the mat. So we wanted to see what the most abundant members of the iron mat are doing. and it turns out that there's a lot of sulfur and thiosulfate oxidation. We have hydrogen oxidation, iron oxidation, potentially also some manganese oxidation, with the manganese present in the fluid, and methane oxidation. And we have both aerobic and anaerobic capabilities, it seems. So these iron mats kind of support the carbon, sulfur, nitrogen, and iron cycling. And we have mostly lithotrophy happening, but we also have some heterotrophy symbionts that we see in this CPR bacteria superphylum. These are very small celled bacteria that are proposed to be symbionts, and members of the group that we have found in the net have been hypothesized to have gamma protobacteria as their hosts, with gamma protobacteria being very abundant in our net. Yeah, but we can see that iron is used by zeta protobacteria in the iron nets. However, iron oxidation could also be performed by other microbes in the nets, such as gamma protobacteria and alpha protobacteria, of the most abundant ones. And here we could see that there are different clusters of Psych2, or the iron oxidation gene. All of them now have in recent years been validated for their iron oxidizing activity by the Clara Chan's lab. And yeah, so we know that Girsea genomes have the ability, show the ability to oxidize iron, but we also see that they have the possibility to oxidize hydrogen that is present in high amounts at fauna. So they have this nickel iron uptake hydrogenases, which is not new for the genus. But we wondered where and kind of when did zeta protobacteria get these hydrogenases. So when you look at the closest relatives, you find sulfur oxidizing gamma protobacteria, which has been found before. And you can see that they often come from microbial mats or the symbiont host connection. So they're like, would be symbionts from animals like clams and shrimps at hydrothermal vents. So we wonder if both kind of host environment or the microbial mat dynamics could kind of support this maybe horizontal gene transfer that would happen between gamma protobacteria and zeta protobacteria in these environments. But yeah, looking at the phylogenetic tree, it doesn't look as straightforward. It is still very likely that hydrogenases in zeta protobacteria were obtained by horizontal gene transfer as kind of an adaptation to these high hydrogen concentrations. And since the kind of energy efficiency that you get from iron is quite low, then maybe that would be like another niche for them, for this group of zeta protobacteria, to get a bit extra energy when there is really high amounts of hydrogen. Yeah, but as with all metagenomic studies, you can be curious if actually these zeta protobacteria really oxidize hydrogen in the environment. And what we have tried to do is some proteomics and some transcriptomics, but as you may know, if you're working with samples with a lot of minerals, they tend to make extraction a bit tricky. So we needed to use something else. And what can be used, for example, to predict expression of genes is codon usage bias. So using different, the way that microbes use different codons can help with bioinformatics methods to kind of predict how much they would get expressed. And we could see that iron genes and hydrogenase genes that would be needed for iron oxidation and hydrogen oxidation were both very highly expressed in Girsea genomes. So this could indicate that they carry both processes in this environment. So that they would do iron and hydrogen oxidation is nothing new. So the isolate of Girsea vivora has previously shown that they could do both things also at the same time, which is kind of a rarity in Zeta protobacteria. It's only this genus that has been shown to do hydrogen oxidation. But when grown in the lab, this Girsea did not produce tubular structures or these sheets of iron oxides, but what they call amorphous particulates, so kind of just particles of iron. So if we have a Girsea representative that makes iron oxide sheets and also oxidizes iron and hydrogen, this kind of opens up new questions if hydrogen oxidation kind of influences iron oxide sheet production, and whether this can influence and is relevant for iron oxide structures as biosignatures or records of early Earth conditions. So this is kind of like a small question that I've been asking myself. Yeah, but there are several sites where iron mats are present at hydrothermal vents. In this place, I haven't really included them all. This is a bit of an older map. But yeah, I'm always also very curious to hear if any of you have come upon iron mats when on dives. But yeah, most of iron mats that were closely studied are like here on the Luigi Seamount and, for example, Mid-Atlantic Ridge and Mariana Trench. Not Mariana Trench, but at the Mariana Trough. And we also have, for example, at Mid-Atlantic Ridge, we have these shrimps, vent shrimps that have, for the first time, shown a symbiosis with Zeta protobacteria. But most of iron mats that were closely looked at were kind of on the seafloor or were on the diffuse venting sides with low temperature and high iron concentration. But at Fovne, we instead have high iron and high hydrogen concentration and mats growing on hot temperature black smoker chimneys that are also very porous. And this geochemistry makes Fovne like a unique study site for iron oxidizing microbes and also their adaptations. So in summary, we see Zeta protobacteria of genus Girsea at Fovne that seem to be producing this iron oxide sheet and forming dense iron mats. And with these iron oxide structures, they also provide the environment for other microorganisms and supporting the carbon, nitrogen, sulfur, and iron cycling in these iron mats. And the iron and hydrogen concentrations at this site make Fovne a unique study site. So with that, I would like to thank my research and the people that are involved in this project, together with my PhD supervisor, Runar Stokke, and so the Deep Sea Biology Research Group and Center for Deep Sea Research. So there will be several publications that will be coming out from the group quite soon. So if interested, keep an eye on it. Yeah, and with that, I'd like to thank you for listening, and I'm happy to take questions that you have.
Thanks a lot, Petra. This was a really nice talk on other player advent. I think we already have some question in the chat from Leon Hoffman. Do you want to me to read your question, or do you want to say it?
Oh, I can read it. Hey, this was
Okay.
again a very cool talk. Many thanks. I've been recently working in the Indian Ocean, and we always see these bacterial mats near the hydrothermal vents, and they're always red. So I guess it has something to do with iron, even though we never really in detail investigated it. Did you examine their use as food source for animal grazers? I know, so the non-symbionts. It's clear that Bathymodeolus and Rimicaris, etc., they use them as symbionts, but there are also many bacteria that I think we see many grazers down there, particularly gastropods, so mollusca, and they must feed on something, and that something is not quite clear to us. You have any idea on that?
Yeah, that's a great question. I wonder, you see these grazers like on the mats that they could potentially like feed on them?
Well, actually, they're quite often covered in bacteria themselves. So they must be in direct contact with the bacteria, so somehow, or the bacteria simply settle on them and use them as a lift mechanism, actually, for them to get exposed to the nearest iron that they can find. Yes. So they're always close to the mats, but whether they actually eat them, it looks to me like the obvious food, actually. But what we saw is that there are, within one genus, you may have several species developing, different species developing, without that it's obvious that there is different food for these species. And that is, it must somehow, well, we have a feeling that it must come from these bacteria.
Yeah, no, I think quite possibly. I think like mats in any case are often very, like, it's a big source of polysaccharides that they expel anyway, like in all types of mats. So I think that could be very a rich food source. In our case, on these black smoker chimneys, we haven't really observed something like super visual. Like, we don't see directly on the mats or closer any like gastropods or something. Yeah, I think that would still, like in the Arctic, a lot of species that are there, sometimes they're very small. So if you just look with the RV, you don't maybe see them. But if you would look more closely, then then we sometimes find like really small snails or stuff like that. But the chimneys have not been like looked at that much. And with metagenomics, at least, we don't get much of the eukaryotic sequences. So here, yeah, I don't know. At least like just a first look at the chimney is looks like more bacterial based, but when you move to more diffuse areas, then you find more eukaryotic life as well.
I can provide an additional remark based on some observation we made on vents in the Izu-Ogasawara arc, and we saw some shrimp, not the Rimicaris exoculata shrimp, but other from the same family, that were directly grazing and eating mats they were collecting on the shell of mussel. So we published, we actually are publishing the video of this observation. That should, like, the paper is in review, but it should be available soon. And but yeah, I think it would be interesting to see if there is between different type of mats. And I think at Tag Venfield, if I remember well, there are different type of mats, some iron mat and some that are more sulfur mat with just Campylobacter bacteria. And it would be interesting to see like if there is some specialist among the grazing fauna, like some prefer some type of mat and some other. So yeah, would need to find a case study. I think the Arctic is maybe not the best place because like there is not so much fauna there, I think.
At least not, yeah, like an obvious here in this case. Yeah. Yeah. But oh, that's good. I would love to see that. Yeah. I think yeah, I mean it's a different food source as well, right? Because then you have so much iron in the mats, and since iron also attracts a lot of, I mean there's a lot of heavy metals normally in the vent fluid, so if it binds all these things, then it might be kind of a bit more toxic to graze on that mat. But of course, I think it depends on on the fluid, if it's already very mixed or not, like if it's diffused.
Because iron can react very quickly with tissue and can like produce some reactive oxidant species in some case, so it can be damageable for animal tissue. So yeah, maybe some species that eat on iron mats, if there are some, like they would need some specific adaptation for that to not damage the digestive tissue. Interesting. future things to explore.
Yeah, definitely.
Does anyone else have a question for Petra? Yeah, go ahead.
Okay, so yeah, so thank you very much, Petra. Very interesting talks. And so I would like to ask, so biogeography and the dispersal of bacteria in Arctic Ocean. And so you find so many bacteria, in Arctic Ocean. And so the many of bacteria so can be found so other southern region, so or many of them so are endemic to only Arctic Ocean. So
yeah, that's a good question. I think now more studies are coming, like looking at the global picture as well. But I think a lot of times what you see is that the differences between groups, it's more about like the geochemistry that is at specific events that is really affecting the community and what species are present there. So yeah, I think similar species can be really present in different places, but really affected mostly by the geochemistry.
Okay, thank you. So, and I have another question. So, did you find macrobenthos communities in your new discovery of Arctic bents?
You mean macrofauna?
Yeah, macrofauna. So small.
Yeah. So, so some colleagues look at that, but it's more of a diffuse venting in that case, on this vent field. And we also saw it's an interesting vent field because you have like this really high temperature places that look like mostly like microbially based, but then if you look at other places, then you also see some small snails. You get I think it was some there was, I don't know how hydrothermally influenced was that, but they definitely looked at sponges, and there were some anemones that we picked on the side on that at least vent field. But then looking at other vent fields, you get a lot, like there is this like really thin tube worms that are present, a lot of polychaetes, different species that there's a lot of work going into that, and it takes a while. But those species that are specific to Arctic as well.
Okay, thank you.
I had also one small question. I think from one of your figure where you're showing like the the potential of each zeta lineage to either make sheets or make those twisted iron particle. So there is this two strategy. And I think there was one lineage from your study site that had the potential, or at least on the figure, that had the potential to do both. And then you say that this sheet or twisted particle, they could be related to the metabolism potentially, and like the ability to use hydrogen in addition to iron. So yeah, about this lineage that were potentially doing both, like could it depend on like the condition of the physical condition? Like on one particular chemical condition, you can do sheets, and on the other is doing like the twisted sheet.
Yeah, I think that's very interesting. I think maybe it's not connected to the hydrogen itself. Like they have looked at, there's another freshwater group of iron oxidizing bacteria that is very like looked at even more, that also makes kind of twisted stalks and sheets that they know who is making them. And then they saw the difference between like the ones that make the stalks and the ones that make the sheets. It was basically in how they were present in different amounts of oxygen, or like if the gradient of oxygen was steeper or not. So that kind of like sometimes you could already look at an environment if you knew like how much oxygen is there or how steep the gradient is, you would kind of guess who would be present. In this case, yeah, they have also looked at, there's this paper by Clara Chan, I think in 2016, where they looked at the kind of architecture of iron mats at Loihi, and they have seen that the sheets were kind of present more in certain areas towards the other. So definitely there's like an environmental thing going on there. But yeah, I was kind of surprised to see only sheets and only this group of only this genus basically of bacteria that was present there, which I mean was before like, oh yeah, maybe they could make that, but since here you have dominance of only this genus, then you can kind of tie it to the sheets, production of sheets. But I think that they oxidize hydrogen might be, yeah, I think it's like a separate thing. Since, yeah, some do this production and some don't. So it could be any one of those.
Okay. Yeah, so it would be related to the level of oxygen. I see. Yeah.
Yeah, it seems to be that way actually. Yeah.
But yeah, it's interesting to see this sheet because yeah, on the, at least for what I know of the shrimp in the Atlantic, we only see the stalk on the symbiont that are within the shrimp, next to the symbiont, and also on
next to the symbiont. Yeah.
Yeah, like we see the symbiont, the zeta bacteria symbiont inside the symbiont community, and next to them usually there is always some sheets or some particle. The twisted one.
You see the twisted ones.
Yeah,
okay.
and we see them particularly on the eggs, because the eggs have a lot of zeta proteobacteria on the surface of the egg capsule.
Okay, that's very interesting. Yeah, I think, is it like because from the symbiont like paper that was published, there was the same Gersea genus that was found there.
I think it was Mari Profundus related.
It was Mari Profundus in your place. Okay, yeah. Yeah, that would, yeah.
Which makes sense, I think.
Then it makes sense that it would do stalks. Yeah, but yeah. Yeah.
Okay. Well, thanks a lot for your presentation.
Yeah, thank you.
if you have any other question for Petra or Takuya, don't hesitate to send them an email. And yeah, I think we are a bit out of the schedule, but that was an interesting talk, and many of us had a lot of questions. So that's the fifth seminar of the seminar series we started last year. This, for the moment, will be the last seminar we will have in 2023. We might take some times off unless some people want to give us a presentation. But we will probably be very busy this year with conference and talk. There will be the International Deep Sea Coral Symposium this year in May. I think the abstract submission is already done, but registration are still possible. And it will happen in Edinburgh, in UK. And then abstract registration are not open yet, but we will have the chemosynthetic-based ecosystem symposium in Brazil this year, which will be between the 14th and the 18th of August. I think many people in the room would be interested in that, since it was really event session today. So yeah, looking forward to see you there. And yeah, have a great evening or morning, depending on where you are. And goodbye.
Thank you very much.
Okay.
Thanks a lot again.
Take care. Yeah.
Okay, so Pierre, so I will leave, this room.
Yeah. Just waiting for everyone to.
Okay, thank you.
Bye.
Yeah.